All right, so we talked a little bit in class about PCR, and what we were talking about is it's a way to really amplify a small amount of DNA and make a large amount and then be able to do experiments and things like that with. So a lot of times when scientists are looking for particular ways to do experiments and they only have a small amount of DNA, one of the things they'll do is they'll kind of amplify this using PCR, polymerase chain reaction, and then they can take that DNA and then do their experiments with it. So what PCR basically is a three-step cycle that's going to bring about a chain reaction of events that's going to produce really an exponential growth of the amount of DNA that you're starting with. So you take this double-stranded piece of DNA, and then you've got this solution, and what you're going to do is you're going to heat it up, and what that's going to do is it's going to break the hydrogen bonds and cause those two strands to kind of peel apart. And then what your mixture contains as well is some DNA primers in there. You've got some DNA polymerase. You've got a variety of different types of things, nucleotides, etc., so that you can now start to replicate DNA. And so these DNA primers get added, and then once it cools, the primers are going to add to or kind of bind with that DNA that's been really broken apart due to the heat. So you cool it down, the primers anneal to it, and then what you get is the DNA starts to get replicated from the heat-stable DNA polymerase. It was taken from an organism, Thermus aquaticus, taken out of a hot spring, and so that DNA polymerase is heat-stable. So like most enzymes in this heat here would break down, denature, etc. The DNA polymerase from Taq polymerase, or from Thermus aquaticus rather, it's called Taq polymerase. What that does is that has the ability to synthesize the DNA strands in that standard 5 to 3 prime direction using that target sequence in those primers, and it can do so at an elevated temperature that would otherwise kind of disrupt the function of the enzymes. Okay, so what you can see here is you've got this target sequence that you're starting with up here at the top, and then you heat it up and you can see that it's going to separate, and then once you cool it down a little bit you can see these primers are going to start to add to that sequence, and then the DNA polymerase molecule TAQ Taq polymerase, what that's going to do is it's going to start to replicate the DNA, and then after a short period of time what you're going to do is you're going to heat it up again, you're going to cool it down, the primers are going to bind, and then Taq polymerase is going to start to synthesize DNA, and then that cycle's complete, you're going to heat it up again, and so this process continues over and over for a few hours. But what you've got now is you can see each time you do this, by the time you get to the third sequence, what you've got is the actual double-stranded target sequence. So this is going to kind of continue on, you know, in this direction, this is going to continue on in this direction. So what you're getting here now is you've got just the fragments, none of the extra stuff here. This is where it's really going to start to ramp up and produce large amounts of DNA, because every time now it gets to the end of the fragment, you know, it's done. It gets to the end of the fragment here, it's done. So you can see here, instead of having these really long sequences that we started with, by the time you get to the third sequence here, you're going to have the actual target sequence that you're after, and you're going to be able to go very rapidly, 1 to 2, 2 to 4, 4 to 8, 1632, very rapidly, within a few hours, get over 1 billion copies of that one DNA copy that you just started with. So typically, you know, you're going to start with more than one piece of DNA. You're going to start with multiple. So you can get large amounts very, very rapidly right from the start. Okay, so what this thing is doing here, what these things are doing, is just kind of explaining what I just had talked to you there. You've got this target sequence, you've got a primer that's been made to it, and then you've got the nucleotides, and then now the Taq polymerase will do its job. Okay, and so when you're looking at this now from the standpoint of doing molecular biology, DNA cloning is really a great way to prepare a large amount of the gene fragment. So what you can do with PCR is you can take that fragment, you can amplify it and make copies of it, and then insert those copies into the particular vector that you're after, whether it's the plasmid, the bacterial cell, whatever it is, and then you can grow those bacterial cells. And what they'll do is they'll do it generally without error. They'll do it much more, they'll do a better job here really than PCR for you to get the gene of interest in a living organism to get amplified very rapidly. So PCR is also used, you know, crime scene investigations. It's used in paternity suits, molecular biology here. So there's a wide variety of uses for it. The main thing to keep in mind really is that it's the method for amplifying a small amount of DNA into a large amount very rapidly. Okay, so here's another video that you can watch. Follow the link from the presentation that you downloaded, and it will also explain, the things that I just explained to you. And certainly if you have any questions about any of this, make sure that you ask and we'll go over it in class, okay? All right, so we'll talk to you soon.